Hello, friends, and welcome back to the channel again. It's always beautiful to have you here, and it's wonderful to discuss our science concepts. Today, we remember we are doing a series on biochemical tests, the use of different biochemical tests to be able to identify different microorganisms using different features that these microorganisms have. Today, we are talking coagulase test. Coagulase test is one of the tests that are used to differentiate organisms. So we say that organisms are either coagulase positive or coagulase negative. And like we inferred in some of our earlier videos, coagulase is an enzyme. So the presence of that enzyme, means that that organism is coagulase positive. And if that enzyme is not present, the enzyme coagulase, then we say that the organism is coagulase negative. Now, it's important to note that this test can help us to differentiate organisms, one from another, especially when we talk about Staphylococcus. For an example, we have Staphylococcus aureus, which is coagulase positive, and we can use that feature to differentiate it from other species of Staphylococcus. You have Staph epididymis and other different types of Staphylococcus which are not coagulase positive. So this is important to know. Then it's important to know that in this coagulase test, there are two types of coagulase that we search for. We have the one we call the bound coagulase. This is the coagulase that is attached to the cell wall of the bacteria. And then we have what we refer to as the free coagulase. And this free coagulase is coagulase enzyme that has been secreted by the bacteria into its surroundings. So any one of them will suffice. Any one of them is good enough to test for the coagulase test to know if coagulase is present in the bacteria or not. Now, when we do the coagulase test, the importance of the coagulase test is that this enzyme coagulase has the ability to help to clot blood plasma, and this is very, very important. It has the ability to clot blood plasma. And what do you see that happens in clotting? When you look at a fresh wound and the wound now heals, what happens is that blood clotting has occurred. And for blood clotting to occur, there are a cascade of events. There are many scientific events that happen to stop the bleeding, and you have uh, a protein called fibrin or fibrinogen, which is largely involved in this process, the process of blood clotting and by extension wound healing. So coagulase as an enzyme has this ability to clot blood. And for us to know if a bacteria is coagulase positive or coagulase negative, we use rabbit plasma. Rabbit plasma is what we use. We test these cells against rabbit plasma, and then if these cells have the ability to bring about the clotting of rabbit plasma, which you would see with your naked eyes in a reaction that we refer to as the agglutination reaction. You would see it clearly on in an agglutination reaction, and then you'll be able to know if the organism is coagulase positive or coagulase negative. And like I said, you can have either bound or free coagulase. And so testing for either of the two, you have two different methods. If you're testing for bound coagulase, you're going to test for that using glass slides. So it's a simple test. Two glass slides, one acts as control and one is the test group. And then you put either saline or distilled water on the two glass slides, and then you put your rabbit plasma, just a drop, maybe 1 ml, 2 ml, you put it on the glass slides that have distilled water or saline on them. And when this has been done, then you inoculate your organism onto the two slides. Remember, one is a control, one is a test. So it is to even the test one that you put your rabbit plasma. The other one just has it just has distilled water. Now, when you inoculate your organism onto the two slides, you just watch. If you see a change in color, clumping of cells, you know that that's an agglutination reaction, and you know that that organism is coagulase positive. If you check, and normally in the control, you should not see any change. So it's easy to differentiate because the control doesn't have rabbit plasma, so it's easy for you to know if the organism is coagulase positive or coagulase negative. That's the slide, and that's what you use to test for bound coagulase. That's the coagulase that is attached to the cell wall of the bacteria. Now, for free coagulase, it's simpler, and like I told you, I said the free coagulase is the one that has been secreted by the organism into its surroundings. And when you have the secretion of this free coagulase into the surroundings, then you have a test. The test is simple, it's a test tube test. So just the simple things too. You have your distilled water, then you have your rabbit saline, and you also have the control group, and then you inoculate this test tube mixture, with your test organism, and then you have the control and you have the experimental or the test group, and then you incubate at 37 degrees for 2 to 4 hours, and you check. If you see agglutination, if you see a change in color, you know that organism is coagulase positive, and if you see no change in color, you know that it is coagulase negative. And then you can wait for 24 hours for the test tube test, the one you are incubating. You can wait for 24 hours to be certain about your results. So these are just coagulase tests, and this is important to know. It helps us to know if an organism is carrying the enzyme coagulase, and if it is carrying that enzyme coagulase, it will have the capacity to clot blood, and this will be seen when you do the coagulase test. So this is just one of the tests that we do in our biochemical battery of tests. We've discussed what the biochemical tests are, and this is one of the different types of biochemical tests. Thank you for listening to this, and just keep your eyes peeled as we go through all the different biochemical tests and exactly what it is we are testing for and how that helps us in the identification of microorganisms. Do have a wonderful day.